Hello, everyone. Welcome to San Diego. This is the home I grew up in, and welcome back to our next video on the signal transduction pathway. So we've already discussed some of the special cases of how cell communication occurs. Now we're going to talk about how it usually occurs in humans. So there's many. You're gonna hear me say the word usually a lot today. Um, that's because cell communication and cell signaling is still something that's on the cutting edge of biology research. And so it's a great opportunity for us to learn more. So I'll show you what is our current understanding of the beautiful complexity which the human body communicates between cells. So last time in the last video, we talked about the different ways that reception occurs. Step 2 is transduction. And transduction just means changing the chemical form of the signal. Typically, this is because there's a ligand which has received the hormone, like epinephrine, on to the intended cell, to the target cell. And that's going to create a cascade of events. Now, there's two main pathways that you want to know about. The first one is called phosphorylation, and that relies on the group of proteins called protein kinases. Your book goes into detail about this. Here's what you really need to know for the AP test, is that it's often a series of reactions in which multiple proteins are activated. So one protein activates a second protein, and even activate a third, fourth, or fifth protein. This is called a phosphorylation cascade. You should know that this is done by enzymes called kinases. K-I-N-E-S-E. Maybe I'll put it in the chat. Kinase. So anytime you see that word kinase, you know that that's an enzyme that phosphorates proteins to make them active. That's good because that allows for simple signal transduction. This epinephrine that bound to the cell is now causing things to happen in the cell. That's how the signal is going to get turned into the response needed in the cell, such as glycogen breakdown or whatever else is needed for the specific hormone. Okay, so let's talk a little bit more about that. Well, an important part of that is also, the next keyword is secondary messengers. In the case of epinephrine, the secondary messenger is a very important one called CAMP. You should study what exactly that is. Of course, it's related to ADP. It's a cyclic version, and it's a direct product of ATP. So cyclic AMP is made by a membrane protein taking ATP, and it makes that. And even though there can be little amounts of it in the cell, when there's lots of CAMP, it activates certain pathways that will ultimately lead to this desired cell response. And what we found that CAMP is not just used by epinephrine, but actually by a variety of hormones. And so CAMP becomes what's called a very important secondary messenger. It's the one carrying on the messenger. And just a quick note, secondary messengers are not proteins. So we have many proteins carry on the message, but those are just proteins. They're always present. The secondary messenger is actually not a protein. It's something your body actually has to make that's not already there. And that allows for signal amplification. So that's one of the great things about a signal cascade is that one epinephrine, or let's say 10 epinephrine, because there's multiple receptors all throughout the membrane. So let's say there's 10 epinephrine that bind to a single cell, or 10 epinephrine, each one are going to make many, many CAMPs, and each CAMP is going to lead to glycogen breakdown. And so, only 10 epinephrine binding to a liver cell could lead to the release of thousands of glucoses. Very cool, very important. It's important that all of these processes are reversible. And so that CAMP ultimately gets turned back into ADP by a different enzyme. And so all of these are temporary reactions which allow for greater regulation. So what is the ultimate goal of this? Well, let me just mention one last secondary messenger that you should know about, and that is calcium. So calcium ions are abundantly present in the cytosol, and so in certain pathways, the specific, let me read it quickly. In certain pathways, calcium is used to broadcast signals quickly, and this is common in G proteins. Yeah, and it's a secondary path. So calcium is another important secondary messenger. Again, by opening it up an ion channel, we can quickly get lots of calcium in the cell, and that can greatly amplify the message and transduce it as well. So what's the point of all this? Well, let's talk quickly about what are the cell responses. Well, there's actually thousands, so we'll just talk about some typical cases. One case is epinephrine, which leads to the cell response of glycogen breakdown. What you want to know in all of these responses is that responses have all have this characteristic. Number one, they are. This is what you need to know about cell responses. Let me turn the page. Okay, number one, they are fine-tuned. So they're fine-tuned. So it's not just on or off, but they're fine-tuned, meaning they can be turned on or off for a very specific amount of time, and they can be amplified or modified in many different ways. So it's not just like a light switch that I could turn on or off. Number two, they're specific to cell types. So epinephrine only will interact with cells that have epinephrine receptors, and that's true of all hormones and steroids. They need receptors. Even something that we talked about, like testosterone in the last example, that could go through the cell membrane. There's only certain cells that have the receptors to interact with the testosterone. The testosterone will just diffuse in and eventually back out of cells that do not have the receptors or the proteins to respond to it. Number three, it can involve scaffolding in proteins, which increase efficiency. And number four, and most importantly, the signal is reversible. So all of these processes all need to be reversible. If they ever become not reversible, that's often the cause of cancer or other genetic conditions. And lastly, two common cell responses that you want to know in detail are gene expression, and apoptosis. Gene expression means that because of this cascade, we're going to start expressing new genes and causing the creation of new things. And apoptosis is cell death, programmed cell death, because the cell is too old, it's injured, or it has become infected, or it could even just be part of embryo development. That's a common misconception that apoptosis is only happening when the cell's sick. And it looks like my brownies are done, so I guess it's time to go. I hope that makes sense. Please let me know any questions you have about cell communication, and have a good day.